So prescribed antimicrobial control. Thank you. Oh, thank you, sir. Thank you very much. Let me go ahead and download that real quick so that I will have it.
Today I'm going to drink.
Okay. Thank you. Yeah. Okay. So when we talk about prescribed antimicrobial control, it's really pharmacological, right? So we're going to be talking about, I've selected 5 antibiotics to talk about. And I'm going to talk about the mechanisms of how the antibiotics work, right? So in the long semester, I would have given you all a take-home portion so that you could have done some of this on your own and learned about the antibiotics, right? It's just not feasible in the summer for lots of reasons. Number one, you've already got the 20-point assignment, and I just didn't want to tack on more stuff. So after Rosa called me and said, Provi, your unit 2 lecture is way too long. I went in and looked at it, and I said, well, it's time to change this up a little bit. So I cut it into 2 pieces, and I took the antibiotic lecture, and I made a new one. Right. So I did this Sunday. I think I did it on Sunday. I made a new presentation just to talk about antibiotics, because they really are very important, and we really are in a crisis. I think James has kind of led on to this, that we're in this crisis of antibiotics because we've overused and misused antibiotics for the last 30, 40 years, right? So just think about it, right? You would go to the physician and you would say, I'm sick, I think strep throat, please give me an antibiotic. And the physicians would look back there and see no indication that it was a bacterial infection. Yeah, that it was a bacterial infection. And so what they would do then is say, well, you don't have any fever. It looks like you have a little viral or a little allergic. You're being troubled by allergies. So there's really not much we can do for you. And then they would get really mad and basically say something like, well, you're the worst physician ever, never going to come back here. And so physicians were really pressured into prescribing an antibiotic when it wasn't necessary, right? So they would say things like, well, I'm going to go ahead and give you this prescription for amoxicillin, but don't fill it unless you start to have a fever, right? And the patient would say, okay. And then the patient would leave the office, and guess what? They would go, fill the prescription, right? And so nowadays we've gotten so technophilic that the very few physicians actually write prescriptions anymore. They just send the prescription directly to the pharmacy, and that way there can be no misuse of the prescription. But that happened for a long time, and then people can go over to other countries like Mexico, and they can buy the big jars of ciprofloxacin and the big jar of augmentin, and then they can just bring those back to the United States and dispense them when they need to, right? And so you can see how the misuse and the overuse. And the other thing is the patients are to blame also, right? So it's not just the physicians or the PAs, but it's also the patients, because the patient, first of all, we like oral pills, right? We don't want to take shots for whatever reason. They don't even hurt that much. But so we get prescriptions that are by mouth, right? It's more profitable for the drug companies, that's true. But we just, as Americans, we don't like to take injections. But so we get them by mouth, and then it says, okay, take this TID for 14 days. Well, guess what? We take it for the first 3 days, and then we feel better, and then Mercy's throwing a party and she's going to serve us margaritas. Well, it says contraindicated by alcohol. And so we, as a patient, me or anybody else, as a patient who doesn't know any better, would just stop taking the antibiotics. They feel better, and you go and you have fun at the party. But then what happens is some of the more some of the bacteria that are there that were being kept at bay but not killed yet now can come back, because number one, when you drink alcohol, you weaken, you suppress your immune system, and because you're not taking your antibiotic, the organism comes back, and now you have a stronger grouping of bacteria that are harder to kill, and so now you're in real trouble, right? And so those things have been going on forever, right? So let's talk about these antibiotics. So the pharmacy folks and the people who teach pharmacology have you all, I think they still have you all do drug profiles, right? But those drug profiles really are just kind of uh, some information that you can that they want you to understand about the drugs that you can take out of the pharmacopoeia or whatever, right? But I would much more be, from my perspective, from the micro perspective, I'm yeah, the side effects and adverse reactions also, but I'm much more interested in the mechanism of action. So when we think about the mechanism of action, remember, antibiotics are only effective against a bacterium, an antifungal against a fungus, an antiprotozoal against a protozoan, and an antiviral against a virus. But there's very few drugs that cross over, right? So here you can see that there are lots of different targets that we've talked about, right? So we talked about the cell wall, and so you can see the penicillins, the cephalosporins, the monobactams, sulbactams, the carbapenems, all those drugs there. They're all targeting the cell wall. Plasma membrane, there are polymyxins, right? And the lipopolypeptides, right? Those things. Then they can target the ribosome. You can see either the large ribosomal subunit, I'm sorry, the 30S ribosomal subunit, small ribosomal subunit, or the 50S ribosomal subunit, the large ribosomal subunit in bacteria, right? And metabolic pathways, right? So you can interfere with the formation of folic acid, like a sulfa drug, those kinds of things, right? Or you can interfere with DNA synthesis, like the fluoroquinolones, right? So you can see, or RNA synthesis, right? But so you can see the different targets that the antibiotics have, and because they're strictly targeting bacterial structures, these drugs do not have an effect on us, right? At least our cells are not the target. If we have an interaction with the drug, there is some component of it to which we are allergic to. That's different. That's a side effect or an adverse reaction, right? But the drug, this is the only grouping of drugs that we take as humans that are not targeted to affect our cells, right? They are targeted to attack the microorganism, the bacteria. Okay. So here you can see are some antibiotics and the spectrums that they actually have, right? You're not responsible for this particular chart. I'm not going to ask you questions over it. But I just wanted to show you that, there is one crossover drug here, a sulfonamide, right? And so it can be effective against bacteria, right? And it also is effective against some protozoans, right? And that the target protozoan for a sulfur drug is Cryptosporidium, right? Which wreaks havoc on our AIDS patients, right? They get diarrhea, and then they have diarrhea for life, right? So sulfur drug helps to mitigate that. All right. So you can see we have very few antivirals. The acyclovir is a good one, and it's been used against herpes for a long time. It doesn't cure herpes, but what it does is it truncates the episodes that we as humans have when we have herpes outbreaks, right? And so you get herpes, either simplex 1, which affects the buccal cavity around the mouth, or simplex 2, which is urogenital. And herpes is a gift of life, for life, right? So you get that from whoever you had sex with, and then you have it forever. When you do have episodes, when you do have lesions show up, you can take acyclovir, and acyclovir will truncate the amount of time that you have to deal with that particular eruption or outbreak. Okay. All right, so I was talking about this. The best way to take a drug is going to be intravenously, right? Because a lot of the drug product will reach the target tissues, right? And so next is intramuscularly, and so you can see. The worst way to do it is, of course, orally, because very little of that drug product reaches target organs or target tissues. That's why you have to take 2 weeks of the antibiotic. Where if you did an intravenous, um, if you had an intravenous drip into the bloodstream, you'd only have to do it once. Intramuscularly, only once, right? They give it into the big muscles of the body, usually the bronchus, right, the butt, your 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 backside, and they give it to you one time, and then that's it, right? Like Rocephin does that, right? If you've ever had Rocephin, all those different antibiotics that you can give intramuscularly, intravenously, there's a lot of them, right? Ciprofloxacin, levaquin, there's a whole bunch of them. Metronidazole. But then almost all drugs are available um, as an oral delivery, right? And Americans like that because they don't have to get injections or they don't have to have an IV put in them, and there's no pain, except that the delivery to the concentration of the drug product that's going to reach um, the target tissues is very little, and so you have to take the drug for, and there's a range, right? For a Z pack for maybe 4 days, but for you know augmentin it might be 14 days, right? So there's a whole range of times that you have to take this in order to keep the concentration of the drug product in the blood high enough that it's going to be effective against the target organism. Okay, good. So what I'm telling you all is that um, if you ever are given a choice, I always want the injection if I have to have an antibiotic. A lot of times it's not available because clinics and physicians' office just don't keep them because it's inventory that they have to deal with, where they could just prescribe and have you take an oral an oral medication, and so that's the way most places do it. But I would always prefer to take the IM so that just take it one time and then don't have to deal with it anymore. Okay. Um. It, yeah, but depending on the type of infection you have, like for instance, Mercy, if you're in the hospital, they're probably going to give you an IV, right? That's going to be the quickest way to help you get better, right? So I know I've told you that I've had my shoulder replaced, and when I had my shoulder replaced, they gave me a prophylactic one one unit of 0.9% sodium chloride and vancomycin. So they gave me that mostly to protect me from not acquiring an infection from the hospital, right? Because the hospitals are, from a microbiological perspective, they're not clean. So they did that and did it one time, and that was it. Yeah, they are more expensive, and so they're not going to be available to us as just people who go into the clinic and say, I'm sick, right? Yeah. No, no, Nelda, they would, uh, from my perspective, and I worked in clinical settings for a long time, um, and from my perspective, people do not want to take an injection, whether it is an IV or or just a needle and syringe. They just don't want it. So I, to me, it's a much better delivery system, but it's never going to come to fruition that we're going to do that because the drug company, it's so cheap to make antibiotics. So the drug companies make a lot of money um, off of the antibiotics, and they're pretty cheap, right? And then they get sold to, there's a lot of, it's very complicated, but it gets sold to a group, and then the group kind of distributes and has clients that are hospitals or clinics or whoever, right? And so there's a markup every time. So let's say the pharmaceutical company sells the drug to this group, right? A hospital buying group. And then that hospital buying group sells it to St. David's, right? Every time there's a markup, right? So let's say they sold it, the pharmaceutical company sold it at a profit to the group for $2. The group sold it to a hospital for $30, right? And now everybody's making profit. Now the hospital sells it and delivers it to the patient for $150, right? So you can see that there is a quite a bit of markup, right? With a lot of the middle people making the vast majority of the profit. Yeah, that's just the way it goes. That's just the way the business is. Okay. But let's just say if you pay $80 for Augmentin in the United States, if you go to Canada or Mexico, if you go to Canada, you might pay $10, and if you go to Mexico, you might buy $10 for a whole jar, right? They're really cheap there because of the way the healthcare systems are set up. Okay. We do want to be leery of potential side effects, right? Toxicity, allergies. So this is called hairy tongue, right? And it happens sometimes. There's Redmond syndrome, and this is a person whose teeth have been damaged by the overuse of tetracycline. So tetracycline biomagnifies in the bones and the teeth. And so some of my friends, I grew up in the 80s, right? And some of my friends, I had a I grew up in a different place than they did. I got bused. Um, I got moved into another school from where I lived, right? The idea, of course, was to have people interact with different ethnic groups. But I had a lot of really cool friends who were very wealthy, and a lot of them had no pimples at all because they were on dosage of tetracycline for a lot of their teenage years. And so some of my friends have these or had these kind of um, discoloration of their teeth because of tetracycline. So they have veneers over their teeth to hide how their teeth really look, right? And so this is one of those things that happens when you take tetracycline for a long period of time, right? Tetracycline has kind of fallen out of favor. It's a drug that's not very often used anymore just because it biomagnifies in the bony tissues, right? So these are some of the things we need to consider when we're using these antibiotics. Other things are, of course, that if you take an antibiotic, it's going to destroy all of the cells that are susceptible to it. So it's going to knock out a lot of the population of the intestines and a lot of the populations in the vagina. And so those are the two most common areas that are affected um, by antibiotics when we take antibiotics. And so because of that, a lot of people in the hospital when they're taking antibiotics because they're sick, they might develop C. diff, right? Antibiotic-associated diarrhea, because a lot of the normal inhabitants, the microbiota, the microbiome have been disrupted, and now C. diff has all this place to come back to. C. diff is a problem because it produces endospores, right? So the endospores are not affected by the antibiotic. And then likewise with vaginitis, right? A yeast infection. Again, ladies, you guys, ladies, you all would know this, but you're more susceptible to a yeast infection after taking a regimen, a treatment um, of antibiotics, right? Because it destroys all of the bacteria that are living in the vagina, and it doesn't affect the yeast because the yeast are not bacteria, and so now the yeast have all the space to come back to, and so that's when you can develop a yeast infection. Anything that causes a disruption in our bodies and reduces the amount of microorganisms that are there, they're keeping each other in check. Um, will allow for other microorganisms to take over and cause problems, right? It's not very common anymore, but douching is also one of those disrupting processes that can happen, that the normal inhabitants of the vagina get disrupted, and then therefore you can have a yeast infection after that. Okay. Any questions? All right. So the key to the toxicity really is the mechanism of action. How is that antibiotic going to work, right? And so I think I showed you my little book that I have, right? The emergency room antibiotic guide, right? And so I have that little book, and a lot of physicians have their own, right? But what you can do is just if somebody comes in with certain symptoms and you say, okay, you've got enteritis, right? You've got an infection of the small intestines, and so you look it up, and you can tell which drugs or which antibiotics are most likely to work, right? And you'll ask the patient, are you allergic to any drugs? And if they don't say they're allergic to any drugs, then onward you give them the antibiotic, right? So remember, bactericidal will kill the organism. Example of that would be beta-lactams and vancomycins, right? Fluoroquinolones, aminoglycosides, metronidazoles, right? There is some discussion about the fluoroquinolones, because in some cases they just inhibit, but in a lot of cases they die, and that'd be things like Cipro, Levaquin, there's Avalox, there's a bunch of them, okay? Others that are bacteriostatic are going to inhibit the growth and require the aid of the host defenses. And remember, the process here is to stop them from growing, and therefore require the aid of the host defenses. Now the immune system, the T4 helpers, can call in and recruit other immune cells, right? Macrophages and neutrophils to come in and destroy the organism because they're not growing anymore, right? So sometimes you can have a relapse of the infection if we didn't take care of the infection with the antibiotic and the immune system, but that's pretty rare, right? So sulfur drugs and chloramphenicol, drugs like that. You do not need to know the drugs that are on this slide, right? I have selected 5 of them that we're going to talk about, and those are the ones you'll be tested about, okay? All this other stuff is just I'm setting you up to understand how antibiotics work, okay? So lots of different ways to remember these things. If you are going to be a pharmacist, then you're going to have to know these things like I do. But again, these are just kind of mnemonics on how people remember different antibiotics that are either bactericidal or bacteriostatic. You all are not responsible to know these, right? So mechanism of actions, you can see, you can inhibit protein synthesis, you can inhibit the cell wall, you can inhibit folate, right? Folic acid, you can inhibit DNA replication. So there's lots of different things that can be targeted, right? And each antibiotic has a specific target, right? You don't take one and it's gonna have all these different targets, right? Now you can take 2 different antibiotics, right? And in some cases, in some infections, like with plague, pneumonic or septicemic or bubonic plague, sometimes we give 3 antibiotics because that's a very aggressive organism. And so we give one that attacks cell walls, we'll give one that attacks protein synthesis, and we give one that attacks DNA synthesis, right? And so you're taking these 3 drugs, but that is the way that we can be sure that we're going to make our patients survive. Okay. So I've selected 5 drugs, and these are the ones that you're going to be tested on. And the stuff that is in red is the most important stuff. Okay. So I've color coded them for you. So the first one that we're going to talk about is augmentin. Now, augmentin is penicillin, amoxicillin, right? And it attached to it is what we call clavionate, right? So here's clavionate right here. This is how you spell clavionate, Right? It's going to be multiple choice, so you're not going to have to spell these things, right? But it's a combination of these two things. These two things put together are synergistic, right? Now, let's talk about the way it works. Amoxicillin blocks the cross-linkage of the formation of the peptidoglycan. So it doesn't allow the NAM and NAGs to attach to each other, right? And so that makes the cells really weak and they die, right? It's bactericidal, they die. There are many organisms out there like MRSA, right? Methicillin-resistant Staphylococcus aureus. The methicillin means that it's resistant to penicillins, to sulbactams, and cephalosporins. And the reason that the MRSA is resistant to these drugs is because they produce an enzyme that's called beta-lactamase, right? So beta-lactamase will go in there and not allow penicillin to work. That enzyme will destroy penicillin. Are you with me? So the way we get around it and to use it for things, not necessarily MRSA, but other organisms, is that we attach a beta-lactase inhibitor to penicillin, right? So we attack a beta-lactamase inhibitor to penicillin, and that beta-lactamase inhibitor is clavionate, okay? So the mechanism of action here is there are two things going on. Number one, there's clavionate, and clavionate inhibits beta-lactamase, right? Now penicillin can work, and penicillin works by inhibiting the formation of the peptidoglycan. It interferes with the cross-linkage of the peptidoglycan layer, okay? That's it, done, okay? So one antibiotic that will attack the cell wall, right? Okay? Questions about augmentin. And a lot of times you'll see augmentin and then it'll either have a little forward slash and have CA after it, and that means it has clavionic acid, okay? This is one of my favorite antibiotics, and my company's made it. We're one of the first companies to design it and sell it on the market. And we came up with bubblegum flavor, and that is so good. So you have to be really careful when you have a suspension of this stuff given it to kiddos, because that bubblegum flavor tastes really great, and kids think it's candy. So you just have to be real careful with it. Okay? Questions? All right. And so here's kind of the difference between amoxil is just amoxicillin, and then you have augmentin, which has amoxicillin and clavionate. Okay? Yes, Lucia?
Are we going to need to know the antibiotics, the SIG, like how they give it, where it said
No,
dose
no,
500 TID?
no. You're not gonna
Okay.
have to know the dosage. No, you're not.
Okay. Thank you.
Just what's in red, okay? Just what's in red, okay, Lucia? Just what is in red.
Okay. Thanks.
That's it. Yes, Angeles. Yes, ma'am. Angeles. I guess she did it kind of by mistake. I guess I don't know. Okay, let's go on. Next drug is Bactrim, also Septra. Right, this is a sulfa drug. It's bacteriostatic. It's synergistic. Right, they're two drugs. I don't necessarily need you to know that one of them interferes with an enzyme and one of them interferes with the binding. I just need you to know that that they are structural analogs of para-aminobenzoic acid. And what that means is they look just like PABA, right? Now, PABA is important because you need PABA to join with tetrahydrofolate to form folic acid. And folic acid is necessary to produce DNA and RNA, okay? Therefore, I've kind of made this really easy to understand. Basically, what I'm saying is that there are structural analogs to PABA, and therefore they interfere and block two crucial steps in the formation of nucleic acids. Okay, that's it. That's what you need to know. So the sulfa drugs are bacteriostatic. They do not allow for new nucleic acids to be formed, and therefore the microorganisms are inhibited. Okay? Any questions about a sulfa drug? The most common one is Bactrim. There's another one that's called Septra that you can get also. There's a lot of other ones, but Bactrim is the most common. Okay? Any questions? Okay. Yeah, sure. So somebody's got their mic on, and I'm hearing, yeah, Angeles, maybe your mic is on, but I'm hearing some kiddos in the background. All right. PABA is para-aminobenzoic acid. It's this thing right here, PABA. Okay. So I'll be happy to repeat it. So the sulfa drug, like Bactrim, they look very much like PABA, and they have a higher affinity, right? They like to join with tetrahydrofolate, right? So they join with tetrahydrofolate and they block PABA from joining with it, right? So they're analogs, right? They look like them and they act like them. The difference is a sulfa drug attaches to tetrahydrofolate very easily, right? Even easier than PABA does, right? So that's a structural analog, right? That's what you need to know. You don't need to know all that other stuff I talked about, right? That's important for somebody like me. But for you guys, you just need to know that it looks like PABA, right? And therefore, it binds to tetrahydrofolate and it blocks the formation of nucleic acids because folate is needed in order to form DNA, right? And RNA. So it blocks the process of forming DNA and RNA. So it inhibits the growth of microorganisms because there's no protein synthesis and there's no DNA replication. So the cell doesn't grow and the cell doesn't produce any new proteins. So it's just sitting there, and the immune system, the macrophages, the neutrophils can come in and destroy the organism. Okay? Any questions? All right. Next drug is a fluoroquinolone, and I've selected Cipro, although they all work the same way, right? But Cipro, everybody knows about it, right? So there is some discussion, some academic and medical discussions about whether it's bacteriostatic or bactericidal. For some organisms, it's bacteriostatic. For some, it's bactericidal. So I probably am not going to ask about that because it's too wishy-washy, right? But what I will ask about is its mechanism, and its mechanism is that it blocks the function of DNA gyrase and topoisomerase, right? Now, those particular enzymes, what they do is they stabilize the DNA double helix when it's being replicated, right? So the double helix is very tightly coiled, right? And so when it unravels to get copied, it wants to go back and be coiled. But the enzymes gyrase, DNA gyrase, and topoisomerase stabilize it so that it doesn't go back to being coiled, and therefore helicase can unzip it and then it can be copied. Okay? So that's what I need you to know. It's going to inhibit DNA synthesis because it doesn't allow for the enzymes gyrase and topoisomerase to function, right? So the DNA never gets unraveled, and therefore it doesn't get copied. Yes, Justin?
Oh, you answered my question. I was wondering if it stopped the DNA from unraveling in the first place, or or did it stop it after it unraveled and it's not able to repackage?
It's a little bit of both, right? So it doesn't allow it to unravel. If it does start to unravel, the DNA goes back to being coiled, right? Because the gyrase doesn't keep it stable, or the topoisomerase doesn't keep it stable. Okay.
Okay.
Good. Yeah. That's it. That's the mechanism. It's a beautiful drug. This is one of the most powerful antibiotics that we have currently, right? But it's being overused. So there's already organisms that are developing resistance to it. And so we just have to be careful about being good stewards to our antibiotics. Okay, because if we're not, we're already in a crisis, right? But if we're not careful, we're not going to have any antibiotics left, right? It's that serious. Okay, questions? All right. Next drug is tetracycline. I selected this because I had talked about it earlier when this stuff biomagnifies in the right. So it's really easy. It's bacteriostatic, and it binds to the 30S ribosomal subunit, the small ribosomal subunit, and therefore it interferes with protein synthesis. New proteins are not made, and then therefore you can see that the cells don't grow, and then therefore they can be destroyed by the immune system. Okay. Any questions? All right. And the last one is rifampin. And rifampin is a very powerful antibiotic. It can be used in lots of different on lots of different organisms, but we mostly use it for tuberculosis because we want to keep the drugs that we use for tuberculosis from just being used anywhere, because then we're going to lose the efficacy of the drug really quickly. So rifampin is typically used to treat tuberculosis. There are some really interesting side effects with rifampin in that all of your secretions of the body will be orange. So you'll urinate orange, your saliva will be a little bit orange, and your tears will be orange, right? It's bacteriostatic, and what it does is it binds to an enzyme that allows for messenger RNA to be made, right? And that enzyme is called DNA-dependent RNA polymerase, right? Polymerase is what makes the RNA molecule from DNA using DNA as a template. That's why it's called DNA dependent. DNA has to be there in order for this RNA polymerase to make messenger RNA, right? So it blocks the formation of messenger RNA, and therefore it blocks protein synthesis. Okay? Questions? Okay, those are the 5 I've selected. Those are the only 5 that you will be tested over. Okay? Any questions? All right, so we are, as James has told us last time, we are in a pickle. It's self-imposed, right? Self-inflicted. We just overused and misused antibiotics, and now we have all of these resistant bacteria, and that's just talking bacteria. It's not talking resistant protozoans. It's not talking resistant fungi. It's not talking resistant viruses. We're just looking at bacteria, because remember, in your health practices, the bacteria and the viruses are going to be the most dangerous to you all, with the bacteria being the ones that cause a lot of problems in a hospital setting, right? Because they're everywhere. That's not to say viruses aren't everywhere, but we're more likely to transmit a bacterium to one of our patients than we are a virus, okay, in the hospital setting. And so you can see you're not responsible for knowing. What you need to know is that we have a big problem, and that big problem is there's a lot of resistant microorganisms, bacteria, and it's because, as James put it so eloquently last time, it's because of our misuse and our overuse of antibiotics. Okay, so this was taken out of the Wall Street Journal, right? It's a reputable source, right? So here are some of those organisms, right? And so what we're going to start today is we're going to start to look at biochemistry in the lab, so that we would be able to identify some of these organisms, right? And we're going to be talking about that in the lab. But you can see here's some of the more important drug-resistant antibiotics in the United States, right? Methicillin-resistant Staphylococcus aureus, vancomycin-resistant Enterococcus, vancomycin-resistant Staphylococcus aureus, and multi-drug-resistant tuberculosis. There are others, but these are the ones that are most important from my perspective in the United States. There's a lot others that are all over the world, and they're here also, but not. These are the ones that cause troubles for us all the time. Okay. How about if you know this one for the exam and only this one, because that's the most important one. So you need to know what MRSA stands for, okay? And what that means, right? MRSA means it's resistant to all penicillins, okay? Let's just leave it at that. All right? Questions? Methicillin is a placeholder for all penicillins, okay? Good. All right. So how do these organisms develop drug resistance, right? Well, it can happen naturally, right? Every 10 to the 8th division, there's mutations that occur. And so these mutations could basically make these organisms so that they are resistant to antibiotics, right? There's also the transfer of plasmids, and those plasmids can be transferred in which group of organisms, Nelda? Which group of organisms transfer plasmids to each other? Do you remember? Mercy? Gram negative. Very good, Mercy. Very good. Gram negative, right? So you have these plasmids being transferred that code for the resistance to, that's okay, Nelda. You have these plasmids that are being transferred to these other cells, and they then become resistant to, say, penicillin, right? Because the plasmid is a R plasmid. It's a resistant plasmid, and it's resistant to a certain antibiotic. Okay. And then there's a lot of other ways. Right? You don't need to know these, right? But I'm just telling you that we have a big problem. And the problem is that the microbes, the bacteria, are just sharing. They're becoming more resistant, and they're doing all these different things because of a lot of mechanisms that they have, right? And so you can see that they have enzymes that they can destroy certain penicillins, like beta-lactamase, right? They can be slow to allow that drug to enter the cell, right? They might not. They might have other enzymes that won't allow the drug to bind to what it needs to bind to, right? They can pump the antimicrobial out of the cell before it can act, right? So there are all these different things, right? And with tuberculosis, right, it produces a special protein that binds to DNA gyrase, preventing the binding of the fluoroquinolone. So you see all these different mechanisms that these bacteria have developed, right? Because we've taught them, right? We've overused and misused. We've allowed them to come in contact with these drugs over and over again. And so they have adapted, they have learned how to resist these antibiotics. And so we are in a problem, right? So we have what we call super bugs, super microorganisms that are resistant to a lot of antibiotics. Okay, so we need to be sure that we educate our patients to be sure that if they're taking an oral antibiotic, that they take the entire dosage of the antibiotic, so that we can be sure to kill off all organisms and not just the really, really weak ones, right? So that's the biggest thing, right? And if when possible, if we know we're going to have a problem, we might use antibiotics in synergy, right? So that we combine two different antibiotics and then give them to the patient, and therefore we can get rid of that particular antibiotic. I'm sorry, that particular microorganism using those 2 antibiotics, so that we're sure that our patients are going to be safe and survive, right? We need to limit the use of antibiotics. Don't give them to people who have viral infections. They're not going to help, right? And we have a bunch of new variations, but they're just not being put on the marketplace because it's too expensive, right? And then we need to search for new ones. One of the coolest things I read recently was that there are some scientists studying the male beard, because there are lots of different molecules in the beard that are antimicrobial by nature, right? Which is pretty interesting to think about. Why not the hair of the head and why the beard? Well, I don't know that answer, but there are people studying the human beard to see if they can find new molecules that maybe they could use to fight different organisms, right? There's a bunch of them out there that people swear by that are like, yeah, I don't know, but there's a whole bunch of different molecules out there that people swear by, right? We talked a little bit about curanderos, right? They use a lot of herbs, right, to fight infections. Also, they have um, essential oils, right? And essential oils have been used for a long time. Has anybody ever heard of thieves' oil? Yeah. So thieves' oil used to be used by physicians who were treating bubonic plague patients, and they would put thieves' oil on their skin. And in that, I don't know if you they wear these masks that they used to wear this mask that kind of had a little beak, and at the very end of that mask, they had some what we would call cotton these days, but they would soak that cotton in thieves' oil, right, which has a lot of different essential oils, and that thieves' oil was supposed to stop the problems from coming into the bodies of those physicians, right? So it's pretty cool to think about. Now, I do know that some of these things work. Um, well, it was mostly it was kind of a what we would call cotton. They called it something different back then. And then yeah, so no, I've never heard that before, James. But the of course that oil was actually came from a lot of different plants and herbs. But they're like, I don't know, there's a whole bunch of stuff in there, oregano, there's a whole bunch of stuff in there, right? I have a container of thieves oil, and I will be happy to show it to you when I get my camera working again. Okay, yeah. So but I can tell you that if we were in the lab, later during the semester, right, we would have done susceptibility testing, and I would have let you brought in your own essential oils so that you could have tested it against Staph aureus or E. coli, right? And what I will tell you is the ones that work um, most effectively are peppermint oil, works really great. Um, clove works. Let's see, what's the other one? Tea tree oil works. And so there's quite a few others that work. Now, I am not a physician, and I am not giving you medical advice. I'm not saying if you have an infection, you should put peppermint oil on it. That's not what I'm saying. What I'm saying is that there is some anecdotal evidence that I have generated that shows that these essential oils work. I have peppermint oil. I don't use it on um, my skin and mucous membranes. What I do is I put it in my sink and in my shower so that the area smells kind of like Christmas all the time, because I like that smell. And so that's what I use it for, right? But it does have antimicrobial properties, right? Just like garlic does, a bunch of other things also. Okay, so that is the end of the material for prescribed control. I called it prescribed control because these are drugs that you would have to get a prescription for. And so are there any questions about what we just talked about? Okay. So the only other course content that's going to be on the second exam is DNA and RNA biology. And I thought that I would go through some of the slides. I'm not going to go through the entire lecture. You have that lecture. I hope you watch it. I'm going to go through some of these slides that I think are really important for you to know. And I'm going to go through them pretty quickly. You can ask questions if you have questions about the DNA and RNA biology. But we're going to have a review on Monday, right? So if you haven't watched the video, please watch it this weekend sometime. And if you have questions about the DNA and RNA biology stuff, then you can ask on Monday as we get ready for our second exam, which is scheduled for next Wednesday. Okay. So any questions? Did you, for those of you who have seen the video, do you have any questions that I can address now? I'm not going to go through everything, but I'm going to just go through this, and I'm going to select certain slides to talk about so that you'll know they're really important to know. Okay? All right. So when we talk about nucleic acids, right, there are two types of them. They're DNA and RNA, and DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. So if we think about DNA, right, it's typically double helix. It's held together by hydrogen bonds. Remember from other courses you may have taken, anatomy and physiology, 1308, 1406, the hydrogen bonds are not really bonds, they're attractions. And so one hydrogen bond is really weak, but a bunch of them together are really strong. So DNA is really strong, right? It can remain in the environment for years, right? So there are cases right now that people who've been sent to jail in the 70s or 80s where they had a little bit of some specimen, either semen or blood or whatever, on either a knife or a piece of clothing or something like that. They're now analyzing that, and in some cases they're exonerating people that have been in jail for a long time as not being the person who committed the crime, because our DNA, you can't run from our DNA technology anymore. It is so specific, right? And so it's important that we understand that DNA can remain in the environment for a long period of time. Hence, if I knew Justin really well, and I really like Justin, and we have the technology to do this now, I could follow Justin around if I was stalking him, and he drank a little cup of coffee, and I could go over and I could get his cup of coffee, and I could collect his DNA from that cup of coffee, and I could make my own Justin if I wanted to. Are you with me? Yeah, that's how far we've come with DNA technology. We can clone people now, right? It is possible. Okay. RNA is important, and RNA is there are lots of different types of RNA, over 22 different types of RNA. In this particular course, you're only responsible for 3: messenger RNA, transfer RNA, and ribosomal RNA, and they're all involved in the formation of proteins. Okay. So if we think about them, then the 3 types of DNA that you're responsible are mRNA, tRNA, and rRNA, right? So messenger RNA is responsible for making a copy of DNA and RNA language. And what I mean by that is that in RNA, there are no thymine, right? Thymine is replaced by another nucleotide called uracil, and I'm going to talk about that in just a minute. Right. So the formation of a messenger RNA molecule from a DNA molecule, that copying is called transcription, right? And it happens almost the same way in the copying happens the same way in prokaryotes versus eukaryotes. How it gets to the ribosome is a different matter, right? They're very different, but how it gets copied from DNA to messenger RNA is basically the same way. It's all done enzymatically, and we're going to talk about some of those enzymes in a minute. Okay. So messenger RNA, its role is to copy DNA in RNA language. We call that transcription, right? Transcription means to copy. Okay. Transfer RNA, once it looks at the information in messenger RNA, its job is to translate that information and to take the information that's in messenger RNA and in a codon, right? Can anybody tell me what a codon is? What is a codon? Can anybody tell me what a codon is?
It's a triplet of nucleic acids.
Of nucleotides, not nucleic
Oh,
acids, but nucleotides. Right. So it would be, for instance, AUG. That's right. And each codon is going to code for an amino acid, right? And the way that transfer RNA knows which amino acid to put into the sequence is because it reads the codon and it has an anticodon, right? So it's codon, anticodon, right? Anticodon is found on the transfer RNA, the codon is found on the messenger RNA. So for instance, if the codon in messenger RNA was AUG, right, that would be the codon, then the anticodon would be what? What would be the anticodon? UAC, that's right, because the base pairings are that adenine always binds to thymine in DNA, and in RNA, adenine binds to uracil. That's correct. And then guanine always binds to cytosine in both DNA and RNA. Okay, perfect. And that's right, Mercy. So the Mercy's had me for 1406. She knows all this stuff. But AUG is always the start codon, right? And so anytime you see AUG, that means protein synthesis is about to start. Okay, so once that series, that chain of amino acids have been put in place, then ribosomal RNA causes the dissociation of the complex, and it starts the folding of the protein, right? So it's involved in making the protein fold so that the protein becomes active. Okay, so protein synthesis. Okay, the rules are. So all nucleic acids are composed of a 5-carbon sugar. In general, it's called pentose, but it's different, right? So in DNA, it's deoxyribose, and RNA, it's ribose, right? It has phosphates, and then it has nitrogenous bases, and those nitrogenous bases in DNA are purines. That means they have 2 nitrogenous rings, right? 1, 2, 1, 2. And they're adenine and guanine. And then the pyrimidines have 1 nitrogenous ring. And so 1 ring, 1 ring. And then here you can see that uracil is an RNA, right? So in DNA, it's adenine, guanine, cytosine, thymine. And in RNA, it's adenine, guanine, cytosine, and uracil. Okay? And the rules of the base pairing are that adenine will always bind to thymine, always, right? And guanine will always bind to cytosine, okay? That's in DNA, right? And RNA, it's the same thing, but there is no thymine. So everything's the same except adenine, binds to uracil. Why do I have 2 lines in between adenine, thymine, or adenine, uracil, and why do I have 3 lines in between guanine and cytosine? That's right. What kind of bonds, Lucia? What kind of bonds? Not covalent. Not covalent. These are hydrogen bonds. No, these are hydrogen bonds. These are hydrogen bonds. Okay. So what that means is that the adenine, thymine, adenine, uracil are much weaker than the guanine, cytosine, and that's where most of the mutations are going to occur. Okay. So if you remember when we were talking about the control of microorganisms, we talked about UV affecting The pyrimidines, right? We said it's going to cause a thymine dimer, and the reason it causes a thymine dimer is because those base pairings are weaker. Okay. All right. So the backbone, right? The backbone of DNA consists of the phosphates and the sugar. So this is a backbone. And then you have the nucleotides being held together. Again, you can see by either adenine, thymine, 2 hydrogen bonds, or guanine, cytosine, 3 hydrogen bonds. Okay, good. Everybody good so far? All right. So the central dogma theory. The central dogma theory is essentially describes the functionality of DNA and RNA, right? And so this really shows that really nicely. So here you have your parent cell, and the parent cell then divides through binary fission, and you get 2 daughter cells, right? Well, in order to get 2 daughter cells, DNA had to be replicated. DNA gets replicated, and each of those particular cells get an exact copy of the DNA. DNA can also be copied into RNA, right? And then you can have transcription, translation. You can have proteins being made, right? So if we think about this in central dogma theory, the theory of how DNA works, right, is about DNA replication, right? This is DNA replication, right? How it works. And then the second tenet of the central dogma theory is about DNA being used as a template, as a mechanism to be copied to make proteins, right? And so this is the second tenet of the central dogma theory, right? DNA replication this way, protein synthesis this way. And you gotta have both of them, right, in order for the cells to do their thing, right? This other thing over here, recombination, is when the DNA gets altered, right? Now that has happened naturally for a long time, but we now have the technology so that we can alter DNA sequences easily, right? We have been doing it for a long period of time. We make GMOs, and so GMOs to me are wonderful. I like them. I like my tomatoes to be so big that I have to have 2 hands to hold them, right? And a peach also, right? But the naturally occurring fruits are going to be really tiny, right? So the naturally occurring tomatoes and the peaches are going to fit in 2 fingers, right? That's how small they are. So almost everything that you buy at the grocery store is a GMO, right? Because they've been genetically altered in order to benefit us as humans and have a larger yield, right? And that's the power of recombination, right? What I'm going to tell you now is that we have the ability now to go in using CRISPR technology and go into disease cells and alter those cells so that we fix them, right? And then the person won't have the disease anymore. And we've done it successfully already in 2 people who've had sickle cell anemia. They went in, they edited the genes of sickle cell, they took them out, and they put the edited genes that say you are now a normal red blood cell, and now these people do not have sickle cell disease, right? So just think about that, right? In the near future, and in my lifetime, I have about 20 good years left, we're going to be able to cure cancer, right? It's going to be expensive at first, but at some point, it's going to be so easy that somebody develops cancer, and we're going to be able to go in there and either insert a genetic code that says, cancer, thou shalt die, or we can say, cancer, you're not really cancer anymore, stop growing, or when you produce cells like this, and we make those cells normal, right? So that's the importance of being able to edit genes, and I think it's a really good thing. We just have to be careful that we use it correctly and do not get mesmerized by the science or enchanted by the money that can be made, right? We need to remain focused and grounded so that we help people and not just try to make a lot of money. Okay. Reverse transcriptase is an enzyme that allows for RNA to be used as a template to make DNA. And this is only found in a few viruses. The most important virus that it's found in is HIV. And the only reason I put it in here is because I don't want you to not take a microbiology class and never have heard of reverse transcriptase. Reverse transcriptase is the mechanism by which HIV replicates, right? It uses RNA as a template to make DNA. Usually, DNA is used to make DNA, and therefore the cells divide. But with a virus like HIV and feline leukemia virus and a few others, they are retroviruses, right? Retroviruses doesn't mean they come from the 50s. It means that they use reverse transcriptase. And so I wanted to be aware of that. Okay? Any questions? Yeah, James. So there is not that then activates. Yes. So usually, and we're going to talk about this when we talk about viruses. Isha, I think your mouth is on. When we talk about viruses, we're going to talk about and you're taking pharmacology. You're probably going to talk about it there also. Is we're going to talk about the cocktail, and the cocktail uses 3 different drugs, and each of those drugs have a different mechanism to inhibit HIV from continuing to propagate and produce more viruses. That's coming. Okay. Yeah. Okay. So I want you to know about semi-conservative replication. What semi-conservative replication means is that when the DNA molecule is replicated and divides into 2 cells, one of the original strands of the double helix is conserved in each of the cells, so that each of the new cells has one of the original strands of DNA and one new strand that was made from that original strand, right? And so this is a really good way to show that, right? With the blue strand being the original strand, and then the red strand being the new strand. And so these are both these new molecules that have one original strand eventually are going to be the DNA in the new cells. And so you can see the new cells, their DNA is composed of one original strand and one new strand, right? And so that's what we call semi-conservative replication. We believe that happens because if that happens, then there's less opportunity for mistakes to be made, right? And so therefore, it's a really beautiful mechanism, right? And that's all I need you to know about semi-conservative replication. Okay. The steps of DNA replication are all done enzymatically, right? And so we're going to talk about that real quickly. But before we do that, I want you to be aware that there is a leading strand of DNA, right? Oops, leading. And the leading strand always is going to follow the forward direction of DNA, which is 5' to 3', right? The other one is the lagging strand. And the lagging strand goes or runs anti-parallel. It still goes 5' to 3', but it's going in the opposite direction of the leading strand. Okay? So does anybody know what 5' and 3' mean? Do you know what those things mean? The 5th carbon, that's correct. So on the 5th carbon and the 3rd carbon, that's right. So on the 5th carbon of that sugar is where the functional group is attached. And on the 5' end, there's always a phosphate group. Write that down. On the 5' end, there's always a phosphate. On the 3' end, there's always a hydroxyl, right? So there's the hydroxyl right there. I'll make a different color. There's the hydroxyl right there, right? And here's the phosphate. 5', 3'. And if you look at the 5' end, there is the phosphate. and the 3' end, there's the hydroxyl. Okay? And that's how you know which is 5', 3'. Okay? Now, what I want to tell you is on the leading strand gets copied continuously. So it gets stabilized, it gets unraveled, it gets copied, right? Just like that. But the 3' end is going backwards, so it has to be copied backwards. And in order to do that, because of the fact that the enzymes are only going to be able to copy on the 3' end, is that the lagging strand has to be cut into pieces and then get copied. And those particular pieces, those fragments, are called Okazaki fragments, right? And so it gets cut by an enzyme called a nuclease, and then it gets copied, and then it gets put together by another enzyme called ligase. Okay? Everybody with me so far? So you have the leading strand that gets copied continuously. The lagging strand is a little bit more complicated because it has to be copied backwards. And in order to get copied backwards, it has to be cut in pieces, copied, and then get put back together. Okay? So I sent you a video of this so that you guys could watch it, because, you know, you can listen to my video that I have about the lecture, and you can listen to me even talking now, and you might say, Bro V, that's really complicated. I'm trying not to make it complicated. Okay? So here are the enzymes that you're going to be responsible for: helicase, topoisomerase, primase, polymerase. I'm not going to separate them out as pol 1 or pol 3. It's just polymerase, and then ligase, right? There's another enzyme that's not here that's called nuclease, and that's the one that cuts the Okazaki fragments. Okay, so I am going to use this particular image, and I'm going to ask you questions about it. I'm going to ask you which is the backbone. I'm going to label things, which is the backbone and things like that. I'm going to ask you which is the adenine and which is the guanine. You'll have to count the hydrogen bonds and then look at which one's a purine and pyrimidine, right? And we can practice doing that at some point. Okay? Any questions? And then you're going to need to know these enzymes, right? The definition, but also I'm going to use, I've selected this particular image for the exam. Okay? So let's just practice with this right now. So this will be the focus for practice right now, right? So if we're looking at this, can you tell me what the enzymes are here? Can you tell me which is the leading and the lagging strand? So could you be able to tell me those things, right? So how about this strand right here? Is it leading or lagging? It's a leading strand. That's right, Justin. And why do you say that, Justin? Why do you say that?
Because it's being synthesized continuously.
Correctamundo, right? So you can see the arrow, the way it's being synthesized, right? So it started over here, and now it's being copied all the way over here, right? The other one is the lagging strand, and we know it's the lagging strand. Can anybody tell me why it's the lagging strand? Why is it the lagging strand? Because it's in fragments. That's right. It's in fragments. So you can see the fragments, right? So let's talk about the enzymes. So number one is the enzyme that relaxes the DNA molecule so it doesn't supercoil. What is that enzyme? What is that enzyme? Topoisomerase. That's correct. Topoisomerase. That's right. Number two is the enzyme that unravels or makes a replication fork so that the DNA can be copied. Okay, that's helicase. That's right. That's helicase. Very good. Let's see. Number five. So in order for DNA to be copied, the polymerase needs to know where to start. right? So number 5 is an enzyme that puts a little bit of an RNA marker. We call that a primer, so that now polymerase knows where to begin the process. What is that enzyme called? Primase. Very good. So number 5 is primase, right? And it's putting a primer in there. Okay. Let's see. So 4 is actually pointing to the primer, right? 5 is the enzyme. Okay. Yeah. So let's see. Okay. So now, if you look at the thing labeled 6, right? If you look at the object label 6, what is the object label 6 called? Polymerase. It's doing the copying, right? And you know, 7 is also a polymerase. I'm not going to say pol 1, pol 2. I'm not going to do that. It's just going to be polymerase. So I'm not going to ask about 7, but I will ask about 8. So what is 8? It's ligase. It's putting it back together again, right? And so it's a little bit confusing because, you know, 8 is also pointing to this individual fragment. What is the individual fragment called? Okazaki fragment. That's correct. That's correct. Okay, good. I promise you, this is the image I will use on the exam. Okay, so if you can make sure that you had good notes on what we just talked about, then you should have, I'm going to show you this image. I'm just to say what is represented by number 8, right? And you're going to pick ligase. It's going to be multiple choice. Okay, good. All right. Let's go on and talk about protein synthesis, right? So we talked about the 3 types, right, of RNA. This is a eukaryotic mechanism. I'm going to show you this image and ask you different parts of it, right? So for instance, I might ask you, what is the process that is going on here? What is the process that's going on here? And so you would say what? Transcription, very good. What is the process that's going on here? Translation, very good. What is represented by this long strand? What is represented by this long strand? Messenger RNA, very good. What is represented by this little thing that looks like a firecracker? tRNA. And what's represented by these other little, not the amino acid yet, Lucia, but what is represented by these little things that are attached to the transfer RNA, right? Those are amino acids. Very good. Very good. Okay. All right. So let's talk about protein synthesis, right? So it happens in 3 stages: initiation, elongation, and termination. Okay. So initiation is the beginning of protein synthesis, and it always starts with what, Mercy? What's the codon that starts it? AUG. Very good. And then elongation is just the reading of the rest of the codons. And so when you read those codons, or when the transfer RNA reads those codons, it puts a series of amino acids in a sequence. And then we have termination. We have a termination sequence, right? And that termination sequence is UAA, UAG, or UGA. Those are the termination sequences, and basically what happens there is they don't code for an amino acid, they code for stop. The process is over. Okay? So the way this is set up is you have, again, initiation, you have a start codon, AUG, and there it is right there, right? You have this large subunit, right? And then you have this small subunit, They come together. They have some sites. The E site stands for the exit site. The P site stands for the peptidal. That means chain. Peptidal means chain. And the A site stands for acceptor. So the transfer RNA comes in, it gets attached to the anticodon to the codon at the acceptor site. And then when it hits the peptidal site, the amino acids are transferred to form a chain. And then when in the E site, the transfer RNA leaves and goes and does its business, right? But at that ribosome, you're having the sequence of amino acids get put in. But it all starts with AUG. So you should know that AUG is the start codon. Okay? Elongation, it just happens at that P site, right? Where these amino acids get put into chains continuously, right? And then eventually you come to the termination sequences, right? Which are UAA, UAG, and UGA. You should know those, right? You should just know those because that makes your life easy, okay? And so we talked about codons. Once those show up, protein synthesis is complete. It's done, right? So we talked about codon and anticodon, right? And so now how do we read these things? Well, we have tables, right, that are codon tables. And so the way you read this, right, the way you read this is this is your first base pair, right? So let's say AUG, right? Here's A, right? This is the second one, right? Here's U, right? So bring it down here, and then you come over and this is where G is, right? So you bring over AUG, and that codes for methionine. So each codon is going to code for a specific amino acid. So let's practice with some because I'm going to have you do this on the exam, right? So GGU codes for what? GGU codes for what? Well, let's go back here. Let's go back. G, G, and U. So GGU codes for glycine. Okay? You see how it works? So GGU codes for glycine. What about ACC? Right? What about ACC? Well, let's come over here. A, C, C. Yeah. So ACC, right? ACC codes for threonine. So explain what again, James? Help me out with that.
I'm sorry, I haven't watched the video, so this is partially, I guess, my fault. But what did you do to figure out how to use this? I don't understand how to use this table, so that's what I'm trying to figure out.
Okay. Fair enough. So the way you do this, let's just take a codon. Let's take AUG. Okay, James. Here's AUG. So AUG, this is the first nucleotide, the first base in the codon. This is the second, and this is the third, James. Okay. So if we look at A, here we start here, A, but then we go up here and get the second one, U. Right, here's U, bring it down, and then we bring the G over, and we have AUG. So this is 1, 2, 3, and we get AUG, and that is methionine. James, did you follow that? And somebody else, I didn't catch it, but somebody else said the same thing. Did you see that?
Yeah.
James.
Thanks. Thank you.
Let's do another one. Let's do ACC. Right? ACC. So James and Rosa, tell me what ACC stands for. What does ACC stand for?
Adenine, cytosine, and cytosine.
Oh, yeah, you're right. But what amino acid does it code for? What amino acid does it code for?
Threonine.
Threonine. Very good. Right. What about UGA? What about UGA? What does that code for? Anybody can answer. What does UGA code for? Stop. Right. It doesn't code for an amino acid. It just means that the process is over. It's done. Okay. Okay. So if you haven't If you haven't watched the video, please watch the video, and we're going to have a review. So I'm just hitting the most important high points of the video. There's a lot of stuff in there, but I'm hitting the high points, right? The stuff that I know is on the exam. There might be some other stuff that's in this video that that might be on the exam, but I'm hitting the high points. I'm hitting 90% of everything that's on the exam that's dealing with DNA and RNA biology, right? Good. All right. Let's go on. Okay. The last information here is about mutations. Yes, Nelda. Yes, Nelda.
With that being said, will you have that chart in the exam?
Yeah, which chart? Oh, you mean the codon chart? Yes, ma'am,
Yes.
I will. But I tell you what, you are allowed to make your own codon table. As long as it doesn't have any notes on it, I'm okay if you use a codon table. There's going to be very few questions that you're going to need it, and I will include it on the exam when I need to. Okay? Fair enough? Yes, ma'am. Okay. All right. So lots of different ways that mutations can happen. They're going to be one single base pair that's deleted, added, changed, right? You can have a grouping of a fragment. You can have rearrangement of the different nucleotides. It can happen. I don't really care that you know all these ways. What I want you to know is that a mutation is an inheritable change in the DNA molecule. That's all I want you to know. Okay? Good. Some mutations are harmful for us, right? Cancer. Some are beneficial for microorganisms because things like bacteria, they have no way to alter their DNA. So the only way their DNA can be altered by mutations. So mutations are always good for them. They change them up, right? And some can be neutral, right? They have no positive. I'm not going to go into that. For my 1406 folks, mercy, we had a whole half an hour of just talking about mutations. Not in this course. This course is not, I'm supposed to give you the foundation of what DNA and RNA biology is, but not go into any of that other stuff. Okay? So when we talk about mutagens can be chemicals or they can be environmental factors that can change the DNA molecule, right? So they can be acids, they can be UV, they can be lots of different things. For us, the most important mutagen is cigarette smoking. So if you are a cigarette smoker or you live in a household that there are cigarette smokers that are smoking and you're getting secondhand smoke, you're 1,000 times more likely to develop cancer than those who do not come in contact with cigarette smoke. And that includes vaping. Right? So people always say, well, vaping is safer because there's no smoke. No, actually, vaping is more dangerous because you're getting the carcinogens directly as a mist going into your tissues, right? So whatever you heard, if it wasn't that, it's wrong, right? So vaping is even more dangerous than cigarette smoking, okay? So any of these things can happen, right? My father died of gastric cancer, and I believe that the reason he developed cancer was because he was part of a cat tower exposure in the late 80s, early 90s, when one of the cat towers exploded in Corpus Christi. And he was an insurance salesman, and he was in that area when the cat tower, that oil distillation tower exploded, and all those noxious fumes went over this certain area where my dad was at, and that was about 12 hours of exposure that he had because they wouldn't let anybody leave that area. And so a lot of my father's friends and the people in his general age group, a lot of them developed cancer and died from cancer. And I believe that it was because of that exposure to all those carcinogens that happened from the petroleum explosion in Corpus Christi. You can look it up, right? I've sent letters to, yeah. I've sent letters to certain people in the government to try to look into it, but they haven't, and it's been years, so I doubt that they're never going to do that. Okay, questions? But really, when we think about also, we have the ability to use our technology to change a DNA. So it's not that it just happens naturally or that it happens because you get exposed to some environmental factor, right? We have the power to change these things. And we can do that by just inserting a plasmid that we think is important. And that plasmid can be inserted into something like, for instance, I don't know if you know this is talking about a different mechanism, but the one that's really interesting to me is that there is an organism known as Bacillus thuringiensis that produces a crystal, right? And that crystal is not digestible by caterpillars, right? We call that BT, Bacillus thuringiensis toxin. You can buy BT at the grocery store or at Home Depot or something like that, and you can spray it on your tomato plants or whatever. And so when the caterpillars eat that, they eat these bacteria, but they can't digest these crystals. So the crystals, they block up and cause impaction of their intestinal tract, and they die, right? So we've done that already. We can insert a plasmid into corn crops or other things, and so when the caterpillars go to eat those, they don't survive, right? But it doesn't affect any other wildlife. It only affects insects that are going to be chewers and chew up vegetation. That's it, right? So this is one way that we can make a modified genetically modified organisms, a GMO. Okay. So how do these things happen? Well, bacteria are interesting because they're the only organism on the planet that I know of that can take up naked DNA from the environment. So they can take up this DNA from the environment and they can incorporate that DNA into their own chromosome. So they can take up naked DNA, right? They can also when that happens, when the DNA gets put into the chromosome, we call that a transposon, right? So transposon used to be called jumping genes a long time ago because these genes would jump into the chromosome and then stay there forever, right? But that's called a transposon. Okay, now that can happen with other mechanisms also, with plasmids and things like that. Conjugation is a way that gram-negatives share plasmids, right? But the plasmids can then jump into the chromosome, and they can become part of the genetic chromosome of the cell, right? So we have taken up DNA, we have conjugation and plasmid transformation, right? We can do it enzymatically also, right? We have the enzyme systems that can cut things apart. We can insert things. We can do all kinds of things like that. The other delivery system that can happen naturally is transduction. Transduction is a way by which DNA is delivered into a cell by a virus. So a virus, a bacteriophage, infects the bacterial cell. It injects its nucleic acid into the bacterial cell, and then that nucleic acid becomes part of the bacterial cell. Usually that that DNA or RNA that's delivered by the virus basically jumps into the chromosome to take over the cell and make a whole bunch of new viruses, right? But in some cases, the virus might deliver a gene that changes the bacterium to become more deadly. Okay. Oh yeah, exactly. So every single living thing on this planet can be infected by a virus, right? Right. So we use transduction as a mechanism to deliver DNA as part of our transformation processes, right? We can do that scientifically. And so it doesn't need to happen naturally, although it does happen naturally and quite quite effectively and quite normally, right? It happens, but we can do it also. There's a real famous movie that was based on the idea of transduction, of using a virus to deliver. In this case, the scientists just designed a way to cure cancer. They used a virus, and the virus delivered this gene into the cancer cells, and the gene was supposed to say cancer die, right? But instead, the movie goes along, and what happened is there was a mutation. There was a mutation in the gene that was being delivered, and we got a bunch of zombies, right? So does anybody know what that movie is called? Anybody watch that movie? It's fun. No, not Gattaca, but that was a good movie based on genetic engineering. It was called I Am Legend, right? I Am Legend, that's correct. It was called I Am Legend. I Am Legend had Will Smith and Sam the dog, right? And the saddest part of the whole movie is when Sam became a zombie. But it was a really good idea, and it was based on the foundations of microbiology and genetics of using transduction, right? And then transduction went wrong, and we had zombies, right? Okay. And that ends the material exam. Your second exam will be next Wednesday. We'll take the exam, and then after the exam, we'll have lecture and lab. Um, and then after next Wednesday, you guys will go away. We won't have class on the 5th. It's a holiday. The study guide, Mercy, can be due on Friday because I know y'all have a lot going on. So I'm okay with that because quite frankly, I won't validate the grades until Friday anyway. No. Next Friday, James, next Friday. So I really want you all to spend time getting ready for the exam, right? And then, hopefully you get your study aid ready by then so you can use it to study. But I'm going to say that it's not really due until next Friday. You can turn it in early if you want. Lab 3, yeah, I'm okay with Lab 3 being turned in on Friday also, right? I know at this point in the semester, people are like, there's a lot to do. So I'm okay with you guys turning it in on Friday, both the Lab 3 and on the study exam. Okay, good. All right, now I have about 15 minutes, and I'm gonna start the fungi because I don't really have time to waste. This is Unit 3, right? So we'll start the fun. We're not going to get very far, but I'm going to send you a video that I want you. Oh my God, how am I going to do that? So the video is about Paul Stamets, and I would like for you to watch that. It's quite entertaining, and it really will put a lot of the components of my talking about ecology and how the fungi are important in the ecology. But the fungi themselves, there are very few true pathogens. There are only 5 true pathogens in the entire fungal kingdom. But there's a lot of opportunistic pathogens. And so we're going to be looking at 4 of those true pathogens because one of them is only found in South America. And I don't really see a need to talk about something that's only found in South America because a lot of you will never even go to South America. So there's no reason to do that. But I'm going to talk about some of the opportunistic ones also. And so that's going to be, I will also talk about my house I bought and how it is completely infested with fungus. And I'm going to tell you my plans about how I'm going to clean up the house and make it pretty and retire there. Okay. So that's going to be coming. It's always good to be able to use life and real experiences when you're talking about these really nice concepts of science and how they relate to everyday life, right? So we're going to begin to talk about the study of the fungi, which is called mycology. Mycology is the study of fungi. The term mycology is derived from the term mycocities, which means that they have vegetative structures that look like plant cells, but they're not. Okay. So the kingdom fungi, they're all chemoheterotrophs. That means they have to consume some other thing that's either alive or dead in order to acquire nutrients, right? They're all multicellular except the yeast. The yeast are single-celled, right? Their arrangement, the yeast are unicellular. They're single-celled. The molds are filamentous. That means they have these vegetative structures that we call mycelia, and I'll explain what that means in a little bit. And the macroscopic fungi are fleshy, right? And so we'll look at those in just a minute. They're the way they acquire nutrients. Okay, all right. The way they acquire nutrients is absorptive, so they release enzymes to the outside of their cells. They break down those larger molecules into simple molecules, glucose, and then they bring those simple molecules in, and they break them down and use them for their growth and cellular respiration, right? The fungi would prefer to reproduce asexually, like we do, because all of our cells are undergo asexual reproduction, with the exception of our sex cells. And the fungi are much like us, right? They would prefer to produce asexual spores because it doesn't take that much energy, right? And they can do this prolifically, and they do. And so a lot of times people get real sneezy when they are outside and even inside because of the fungal spores that are in the air, and a lot of people are allergic to them, and so then they start to sneeze, right? The fungi will only reproduce sexually when they're under stress, right? And stress to the fungi are no nutrients. So when nutrients become scarce or depleted, the fungi will begin to reproduce sexually in order to produce offspring, progeny that are different, right? The genetic pool is different, which then gives them a greater chance of surviving in a competitive environment, right? They do not form embryos, okay? Good. So the fungi are important, right? Because they are really important in decomposing and recycling nutrients into the environment, right? They form association with the roots and the vascular plants, and they help to mitigate the nutrients from different organisms in nature, right? So they allow different things to be given to pine trees versus oak trees versus, I don't know, whatever else, right? All these different things, okay? The fungi are also used as food, right? There's lots of different foods that I'll show you some of them, right? Besides just eating mushrooms on your pizza or mushrooms in your salad, there's a lot of them, right? They're used in religious ceremonies, right? Some of the syllabins can be used by First Nations people to take them to a higher state of understanding, at least they believe that, and therefore they can communicate with their elders to acquire direction for their people, right? And so this has been going on for years, centuries, right? Okay. We can use the fungi also to manufacture food, right? The fungi produce the first antibiotics. We almost never use them anymore to do that because we can synthetically make antibiotics nowadays. They're used as important research tools because they're eukaryotic, but when compared to humans, their generation period is much quicker, right? So a mold can reach maturity in 3 to 5 days, where we as humans will reach maturity in nowadays about 13 or 14 years, right? Sexual maturity, right? And so therefore they're much better research tool than our cells are because they reproduce much quicker. Now if we compare them to bacteria, they reproduce slower because bacteria only takes them minutes, right? 20 to 30 minutes, right? They cause 30% of them cause disease in plants, animals, and humans, right? Most of them are plant pathogens, right? Some of them are insect or other arthropod pathogens, and then some of them are pathogens to humans and the animals we care about, dogs and cats and cattle and things like that, right? Because they like acidic conditions, they can spoil fruits, pickles, jams, and jellies because those are more acidic by nature. This is a nematode catcher. The fungus itself has these traps, and when the nematode is crawling through the soil, they'll go through these hyphal loops, and these hyphal loops will constrict around them, and they won't be able to get away from the fungus, and the fungus will digest them alive. Isn't that cool, right? So here are some other types of fungi. These are morels. They are delish. I go hunting for them in October. They're beautiful. Some of them have haustoria. Haustoria are appendages that they can stick into cells and steal nutrients from those cells. Almost all the fungi are aerobic. That means they got to have oxygen in order to propagate. A few of them are anaerobic, and that's mostly the yeast. Okay, this is a bird's nest fungus, and in February, I was walking at the park near my house, and it had just rained, and these bird's nest fungi were everywhere. I mean, most people would not notice them, right? You remember that movie, The Sixth Sense, right? So the little boy saw ghosts and nobody else could see them. Well, I see fungi all the time, and most people would just walk right over them and never even notice them. But I see them everywhere because that's one of my things I like to do, right? And the bird's nest fungi were everywhere. I couldn't believe it. I took pictures and all kinds of stuff. Yeah. So you can eat mushrooms, but you have to know what you're doing. Because if you eat certain mushrooms, Mercy, most of them are poisonous, right? So if you don't know what you're doing, Mercy, stick to buying the mushrooms at the grocery store. I wanted to in the horticulture and the continuing ed, I wanted to have a course with my friend. I have a really good friend. Her name is Flo Oxley. She and I go back to grad school together, and we're both mycologists. And so she and I wanted to give a continuing ed course of about foraging in the forest and which fungi were edible and which ones weren't, and what other plants you could eat and things like that. But so far, we haven't been able to do that. Maybe someday. And if we do, Mercy, I'll send you an email so you can take the course. Yeah. Okay. There are 3 different groups of fungi, and those are the yeast, which you'll never confuse because they're single-celled. And they're easily identified because they undergo budding, right? So you can see this little pinching off of the mother cell into a daughter cell is called budding, right? Molds, this is a plate of molds that one semester, my micro class and I put this plate outside of the Riverside campus. And then we let it sit there for about 30 minutes, and we brought it in and we put it in the incubator at 20 degrees Celsius room temperature. And then 2 days later, we looked at it and we could see the mold already beginning to grow. So we let it sit in there because it takes a little bit longer, and let it sit for about 5 days. And we came back, and there are over 200 species of molds on this plate. It's pretty cool, right? They're identified by their spores, right? And that's the easiest. The other way is to do it through an automated system, but that takes a lot of money. If you have somebody like me, you can just look at these things and tell what they are, right? So those are the molds. And then the macroscopic organisms, there are a lot of different ones. Some of them are edible, Mercy. Most of them are not. I have chanterelles that grow in my backyard, and so I pick them and eat them. But you need to know what you're doing, right? Because yeah, they're delicious. But if you don't know what you're doing, then it's problematic. In the green belt, I have other students who go in and collect chicken of the forest, and they collect chanterelles. And so they collect all these different edible fungi, and then they have a fungal feast, right? But you have to know what you're doing, because if you pick the wrong ones, like for instance, you don't want to eat this one. This one is poisonous. This is a stinkhorn. Stinkhorns are mushrooms that emit an odor like poo. Are you with me? So they stink like like crap. Are you with me? And what they do is they trick the insects, the flies and the beetles, to think lunch is being served, and so they come over and they land on this particular mushroom, and they're looking for lunch. And when they finally figure out there's nothing here to eat, then they fly away. But they've done exactly what the fungus wanted them to do, in that they took the spores off of the lamellae, off of the gills. If you were these little things right here, are the lamellae, the gills. And in there are a bunch of spores. And so as the insects are crawling over, they pick up the spores and then they fly off and they land someplace else and they drop off the spores. And that's the way the fungal propagates. Yeah. So yeah, it's pretty cool to think about. Okay. So if we look at these things just a little bit closer, right? Again, the yeast bud. And so for this particular organism right here, you can see this cell. You can see that there's 1, 2, 3, 4, 5, 6, 7 bud scars. So that particular yeast cell has produced a daughter cell at least 8 times, because you have to count that one also, right? And that's pretty cool to think about. These things here are called pseudohyphae. And a few yeasts produce pseudohyphae, and those are the ones that are usually associated with causing vaginitis or oral or anal thrush. And we're going to talk about that in a little bit, right? Maybe not today, because we're about to run out of time. Okay, so yeast. The molds grow so that they have mycelia, right? So if we think about mycelia are the vegetative structures, and there are 2 ways that they form. They can form like this, where they're multinucleated, but they don't have any cross walls, right? And so we call those aseptate. Aseptate. They don't have any cross walls. The other form are more developed, more, if you will, more complex. And these are mycelia that have separations, right? And they're multinucleated. And those things are called septate, right? They're much more advanced. Septate. Okay. So here you can see that these, you can't really tell, but I'll show you in a little bit. Maybe not today, but I'll show you when we talk about this. These organisms are higher fungi. So you can see the spore heads are these little blackened areas. The spore heads are right here, right? You can see them. These are looking them at a little higher magnification, but you can see those spore heads. Inside of those spore heads are thousands of fungal spores, right? And they grow apically, and they grow radially. So this is the center of the fungus, but the fungus is growing out this way, right? It's growing radially. And the fungi all grow radially, whether they grow on your skin as ringworm, or if they grow as a fairy ring, right? Does anybody know what a fairy ring is? Does everybody know what a fairy ring is? I'll show you one in just a minute. Right, so these are the molds, right? The macroscopic organisms are these, like these shelf fungi, these bracket fungi that are growing out of the tree. This tree is in trouble because these are the reproductive structures, but the vegetative structures, the mycelia, are throughout the entire vascular tissues of the tree. Are you with me? And here's a really dainty little one, right? So you just blow on it, you'll break it apart, right? So the macroscopic mushroom fungi are mushrooms, toadstools, bracket fungi, puffballs, stinkhorns. There's a bunch of them. Some of them are edible, most of them are not. Okay, good. Now we're not going to talk too much about the macroscopic organisms because they do not have a lot of clinical relevance. They have pharmaceutical relevance. They have nutritional relevance, but they don't have a lot of, they don't cause a lot of problems from an infection perspective. So we don't talk about them too much here. Okay. So do you remember I said that all fungi grow radially and apically, right? Do you remember I said that? Here are a couple of examples, right? So this is a fairy ring, right? So the the folklore, the legend is that fairies have danced around in a circle, and everywhere they dance around in a circle, they delivered fairy dust, and the fairy dust has fungal spores in it, and so all these mushrooms are growing because of the fairy ring. But really, in for fact, the center of the fungus is right here, and the mycelia are underground, and they've grown radially so that each of these mushrooms are the reproductive structures of the fungus, and they have emerged from the soil, right? So the edge of the fungus is here. It's going to continue to grow, but the edge is right here now. And so all these reproductive structures come up, and they're reproducing sexually so that all these spores get released, and asexually. And so you can see how effective that will be. This is, if you see this, you are quite lucky because, again, if you look at the lawn, it's very healthy. Well, somebody might say it's weedy, probably there's a bunch of weeds, but the weeds, if you will, are really healthy because the fungus that are growing in them are helping each of those plants to be better, right? And so it's really cool to think about. If we think about ringworm, right? This is ringworm on somebody's arm, but the center of the ringworm, the center of the fungus is here, and it is growing radially. Are you with me? In the same manner. And so the other parts of the infection are really like have little pustules, right? You can see where they popped right here. These little pustules have little water, right? And that's where the active growing area is. The fungus will continue to grow unless it's stopped. And it's real easy to stop ringworm. All you got to do is go to HEB or Walmart or whatever and buy a little bit of Mycotin or a little bit of Tenactin or whatever and put it on there, and it'll kill it, right? So these are really important organisms for us to understand, not only from an ecological perspective, but also the problems that they can cause in us. And so we're going to look at them. Okay. I'm going to show you one more slide, and then we're going to stop for the day. And that one more slide is some of the delicious things that you can eat. What causes ringworm growth? Mercy, a fungus. So there are 3 different genera of fungi that cause ringworm, right? And so we'll talk about them a little bit later. Okay. Yeah. Oh, Wilta, how do you know, James? How do you know what Wilta Colche is? Most Hispanics don't even know what that is. Oh, I didn't know that. Where were you, corn smut, that's right. So where did you, what establishment were you associated with? Is this where were you at? Oh, lots of places, okay. Yeah. So, oh, okay, I didn't know that. James, you're gonna have to teach me how to cook, buddy, because I love Huitlacoche, right? Um, and so if you think about it, right, this is corn smut. Most people would just simply basically say, "I'll throw that stuff away." If it's growing and people are throwing it away, I'm picking this stuff up because this is delish, right? And so you can buy it kind of in a can. Very rarely you can buy it at Fiesta, right? Sometimes in a taco, but sometimes you can buy it fresh. And people will pay quite a bit of money for corn smut, right? Huitlacoche. It's delicious. I am so surprised that James knows what that is because almost nobody ever knows what it is, right? I have to explain what it is, right? So this is corn smut. This is chicken of the forest here, and that's delicious also. And then here you can see these dogs are hunting truffles, right? So they find the truffles and then they put them in their mouth. You take them out, and these truffles, they're black truffles, so They're about, I don't know, I guess the market value for these things right now is about $150 for a pound or so. White truffles are a little more expensive, right? So you guys know truffles as chocolate candy, right? And that's just a marketing tool. But truffles really are fungi, and they are quite delicious. You can buy them. A lot of times you might buy truffle oil or something like that. Yeah, it looks scary. Yeah. So, but they're absolutely delicious, and this is just a little bit of the things that you can eat, right? There tempeh, and there's all kind kombucha. There's all kinds of fungal products that you can consume. I know it is because they are just absolutely delicious, right? So I'm gonna leave you with that thought, and next time that we get together on Monday, we will begin to talk about the yeast that are clinically significant, and we'll talk about yeast infections. Then we'll go on and talk about molds and what infections they cause. And then we'll talk a little bit about sick building syndrome and do that. And then we'll begin the parasites, right? And so um, we have a lot of fun things to do left. So I am done with all of the background stuff, right? All of the stuff uh, that is important, but it's not really related to the well, it is related to the microorganism, but it's not really about the microorganism. It's about all the cellular stuff that goes. I'm done with all that. The only thing that we have left to do is later we're going to talk about immunology, but I separate that out completely because I want people to understand the organisms, and I want them to understand the the factors that play in the organisms causing infection in us before we talk about the immune system and how it functions, right? So Unit 3 will consist of the study of the fungi, the study of the parasites, and the study of the viruses. I'm not going to talk about the bacteria individually. I've taken that out. I had to think about how could I put this course and add some time. I'm not going to talk about the bacteria as a group, right? It won't be on the exam. Some of you who were in the course already in Unit 3 might have noticed that there was clinically important bacteria. I've hidden that from you. You can't see it because I've decided that I'm going to cover most of that stuff when I get to talking about infections at the end of the course this summer. And so I didn't really need to give you a little bit of a if you will, a trailer to what's going to come. I just decided to cut that part out and then make the exam about the fungi, the parasites, and the viruses for the third exam. And then on fourth exam, we'll cover um, determinants of health and disease, which is really about virulence factors. We'll talk about epidemiology, and we'll talk about immunology, and that'll be the fourth exam. And then the end of the course is all infection. Okay, so that's yet to come. Does anybody have any questions about anything we've covered today? On Monday, we will finish the fungi, start parasitology. Then we'll go to lab. We'll have lab, and at the end of lab, we will have a review for the Unit 2 exam. The Unit 2 exam will be on Wednesday, and afterwards we have lab and lecture. Okay? Anybody else have any questions before we take a break?